Assalamu alaikum طلاب, and welcome to the first lecture of the histology of the urinary system. For ease of the recording and uploading of this lecture into the internet, we will divide the lecture into two parts, and we will stop the first part by the end of the renal corpuscle, and then we will continue with the second part of lecture 1. The urinary system consists of paired kidneys that are these two bean-shaped organs located on each side of the vertebral column, paired ureters, and single urinary bladder and single urethra. As you know already, the kidneys are responsible for the production and secretion of urine. The ureter will collect this urine and transport it into the bladder, where it will be stored until it will be voided outside of the body through the urethra. The basic functional unit of the kidney is known as nephron, which is this complex structure starting by the renal corpuscle, this spherical structure, and continuing as a proximal tubule, lobe of Henle, distal tubules, and connecting tubules. The function of this nephron is a filtration of a plasma to form the initial ultrafiltrate, which is then pass through these tubules to be concentrated by the process of reabsorption of water and substances from it, and finally it will be formed as the final urine. Our learning objectives of today's lecture is first, to distinguish the key microscopic components of the renal cortex and medulla. Second is to identify the structural components of the renal corpuscle, and finally is to describe the structure of the surface across which filtration occurs. The main function of the kidney, as you know, is the production of urine. And through this important function, we are maintaining the composition and volume of both extracellular fluid and plasma. We also maintaining the acid-base balance in our body, and we are removing certain waste products that might accumulate in our body. For example, if the plasma volume is expanded in our body through ingestion of large amount of water, the kidney will immediately respond by secretion, production and secretion of diluted urine. In contrast to fasting hours in Ramadan, where we reduce water ingestion largely, the kidney will respond by production of more concentrated urine to conserve water in our bodies. Maintaining acid-base balance is another important function of the kidney. As you will learn later, hydrogen ions and bicarbonates are either secreted or reabsorbed in the tubular system of the kidney to keep the pH of our body in certain range. And importantly also, the kidney will remove certain waste products from our body that are toxic if accumulated inside it. For example, the kidney is important, will share in the metabolism of certain drugs to remove the drugs from the plasma, and also very important substances that are protein breakdown products such as urea and creatinine. The kidney will secrete these toxic substances into the urine. These substances, if accumulated in our body, are very toxic. That's why if renal function is compromised in cases of kidney failure, for example, there will be an abnormality in all these functions. For example, the volume, plasma volume will be abnormal. There will be accumulation of extracellular fluid within our body, resulting in edema. The acid-base balance might be abnormal, and there will be accumulation of urea and creatinine in our body. The endocrine activities of the kidney is also very important. The kidneys produce and release certain types of substances that act as hormones, and they have an important functions within the body. An example of this is the erythropoietin that is produced by the endothelial cells of the peritubular capillaries in the renal cortex. This glycoprotein growth factor acts on the bone marrow to stimulate the production of red blood cells in response to decreased oxygen concentration in the body. And that's why patients with kidney diseases or kidney failure suffer from anemia. And in these patients, it is advisable to give them an artificial form of erythropoietin to compensate for the reduced amount of this substance in their body. Another important substance is renin, that's produced by the juxtaglomerular cells within the kidney, and renin is important in the control of blood pressure in the body by the conversion of angiotensinogen into angiotensin 1. Another important function of the kidney is the hydroxylation of the prohormone 25-hydroxyvitamin D3 that's produced in the liver. The final product, which is 1,25-dihydroxycholecalciferol, is the active form of vitamin D3, and the kidney, as we said, is a large bean-shaped organ that has a medial concave border and lateral concave surface. The kidney is surrounded by a fibrous connective tissue capsule, and we can see here in the medial border is the area of the hilum where the main blood vessels and lymphatics enters and leaves the kidney. Also, the ureter will leave the kidney at the area of the hilum. The upper part of the ureter within the hilum is expanded and give rise to 2 or 3 major calyces, and each major calyx will receive the opening of several minor calyces. If we examine a fresh section of the kidney, we can see clearly that the kidney is formed by an outer layer called cortex and an inner layer called the medulla. The cortex within the kidney contain most parts of the nephrons. That's to say, it contains the renal corpuscles, it contains the proximal and distal convoluted tubules. Also, it contains the collecting ducts and collecting tubules. Together with the nephron, an extensive vascular supply to the kidney can be seen within the cortex. If we look closely to the cortex, we can see a series of vertical striations radiating from the medulla into the cortex. These are called medullary rays, and we have in each kidney approximately 400 to 500 medullary rays. These medullary rays are composed of collecting duct in the center and the striated tubules of the nephron on each side of the collecting duct. Between 2 medullary rays, we have the renal corpuscles and its convoluted tubule. And we will discuss this later in more details in the next lecture. The inner part of the kidney is called, as you know, as the medulla. The medulla, as you can see in this photo, is divided into many triangular shaped areas called renal pyramids. And in each kidney, we have about 8 to 12 renal pyramids. This pyramidal shaped area of the medulla is formed by the special arrangement of collecting ducts within the medulla of the kidney. Each pyramid has its base facing the cortex, and it's called the corticomedullary junction, and its tip opened into the minor calyx, and it's called renal papillae. This is the area of opening of several collecting ducts into the minor calyx, and again it's called the renal papillae. Between the medullary pyramids or renal pyramids, we have tissues from the cortex extending down into the medulla, and these areas are called renal columns. Renal columns, and they have the same structure, they have the same tissue composition as the cortex, but because it's located between the medullary pyramids, it is considered as part of the medulla. Further to its division into cortex and medulla, the kidney also divided into lobes and lobules. And as we can see here, this is the cortex, the outer part of the kidney, and this is the medulla, the inner part of the kidney. However, each renal pyramid plus the cortical tissue at its base and surrounding its sides is considered as a single renal lobe. And each lobe will be further subdivided into lobules. The lobules, as we can see here, consist of a central medullary ray and the surrounding cortical material that is composed of different parts of the nephron. And if we can see here, this is a single medullary ray. This extension here, it's a single medullary ray with the cortical tissue surrounding it is considered a renal lobule. Unlike other organs in the body, we do not have connective tissue septa that is separating the lobes and the lobules. We do not have a strict demarcation between the lobules of the kidney and even between the lobes of the kidney. To further understand the histology of the kidney, we should have some points about the renal microcirculation and how blood will circulate into the kidney. As you know, both kidneys receive about a quarter of the blood ejected from the heart. That's to say about 25% of the cardiac output. This is a huge amount of blood, and this blood will circulate into and reach the kidney through the renal artery, which is a branch from the aorta. And within the hilum, each renal artery will be divided into segmental branches. These segmental branches will give rise to interlobar arteries that run between the two lobules of the kidney, that's to say between two renal pyramids. The interlobar artery will then give rise to arcuate artery that run in the cortico-medullary junction. These all are arcuate arteries, and these arcuate arteries will give rise to interlobular arteries. This is the interlobular arteries. So again, we have the renal artery give rise to segmental, interlobar, arcuate, interlobular. So if we go to this photo here, we can see here the arcuate artery running in the space between the cortex and medulla, or the corticomedullary junction, the arcuate artery and the arcuate vein, both together. And we have the interlobular artery. The interlobular artery will then give rise to an important branch called afferent arteriole. Afferent arteriole. This afferent arteriole will be divided into a tuft of capillaries. The first capillary system within the kidney, that is referred to as the glomerulus. Glomerulus. This tuft of capillaries will be surrounded by a capsule called Bowman's capsule, and here is the site of filtration of plasma and the formation of initial ultrafiltrate of urine. Okay? These capillaries will then merge together to form the efferent arteriole. This is the efferent arteriole. The efferent arteriole will then form a second capillary system within the kidney. A second capillary system within the kidney. This is called the peritubular capillary system. These are associated with the proximal and distal convoluted tubules here in this area. This is the second capillary system, the peritubular capillary system within the renal cortex. And from here, the venous blood will start and accumulate to flow back into the arcuate, interlobar, and segmental, and lastly into the renal vein. Okay, so we have the afferent arteriole, we have a tuft of capillaries called the glomerulus, we have efferent arteriole, and then we have the peritubular capillary system. And then the blood will pass into the venous side. Near the medulla, we can see here the instead of formation of the peritubular capillary system, a series of straight blood vessels might pass deep into the medulla, pass straight deep into the medulla, accompanying the lobe of Henle and straight tubules. These are called the vasa recta. These are called the vasa recta, and these are associated with the lobe of Henle. So again, to give you a more illustration of the renal microcirculation, you can see here these branches starting from the interlobular arteries, giving the afferent arterioles, glomerulus, and efferent arterioles. And we can see them here. This is the arcuate artery, giving rise to interlobular branch, and this branch will give rise to afferent arteriole, forming the first capillary system, that is the glomerulus, within the renal corpuscle. Then the efferent arteriole will form the peritubular capillary system, which is the second capillary system within the kidney. And then blood will flow into the venous side. This is all happen in the cortex. In the nephrons that are close to the medulla, this is the medulla for example, in the nephrons close to the medulla, we have another types of branches that either arise from the efferent arterioles or arise immediately from the arcuate arteries, and they accompany the lobe of Henle and collecting duct during their passage in the medulla, and these are called vasa recta. Vasa recta. It's important to say here that this first capillary system that is formed within the glomerulus doesn't share in the process of gaseous exchange between the kidney tissue and the blood. So this capillary system is its only function is to filtrate the blood or filtrate the plasma from the toxic substances and forming the ultrafiltrate or the urine. However, this second capillary system will be concerned with the process of gaseous exchange between the kidney tissue and the blood. And by this, we reach to the end of part 1 of lecture 1 of the histology of the urinary system. We discussed in this part the general structure of the kidney, how it is divided into cortex and medulla, and what do we mean by the renal lobe and renal lobule, what we mean by medullary rays, cortical columns, and other many important terms. We discussed the microcirculation of the kidney, and in part 2 of the lecture we will continue to discuss the histological feature of the nephrons in general, and how it is divided into renal corpuscle and collecting tubules, and we will focus on the renal corpuscle and its component. See you in part 2 of the lecture. Thanks.